Hello, this is Mrs. Popplewell, and we're still in Chapter 10, looking at neurophysiology. Let's talk about nerve conduction. The impulse conduction is velocity and the manner in which the nerve impulse runs down the neuron or nerve fiber. The velocity of that impulse on an unmyelinated nerve fiber means that since it's unmyelinated, the nerve impulse must travel the entire distance of the nerve fiber. So it's very slow. It may be about a half a meter per second. Let's think about that. Think of a meter stick as close to the length of a yardstick, which is 3 feet long, and divide that in half, and that would be about 1.5 feet. So let's maybe 1.5 feet per second. Now, a myelinated nerve fiber is different. It has saltatory conduction. Now, in the picture here, these are Schwann cells, but you would also have myelinated nerve fibers in the central nervous system that is not. In the central nervous system, remember, those would not be Schwann cell. There wouldn't be a nucleus, and there would not be neurolema on the outside. So these are motor neurons in this picture in the peripheral nervous system. So what we're talking about here also occurs in the central nervous system with myelin fibers. It appears that the impulse will jump from one node of Rambier to the next node to the next node, almost jumping from node to node, so it's not necessarily having to travel this distance. Scientists are pretty well unsure of what really happens, but they think that the impulse really just sort of jumps from node to node, so it speeds up the velocity of that nerve impulse. So the nerve impulse can be as fast as 120 meters per second. So for a myelinated nerve fiber, I had a student measure the length of this building that we're in, and it's around 34 meters long. Well, 120 meters, that's almost 4 times the length of the building that we're in. The larger or the more thick the myelination, the faster the velocity of that nerve impulse. So it's very, very fast. So saltatory conduction is faster than you would have conduction on an unmyelinated. Most fast velocity, the fastest velocity are on fibers that are thick and are thick with myelination. Now, we talked about a synapse when we talked about a neuromuscular junction. A synapse is where the nerve impulses passes from one neuron to the next. The synapse is the junction between the two neurons where the nerve impulse is transmitted. It occurs between the axon of one neuron, and here in this picture we have the synaptic knob is attached to a dendrite. So it occurs between the axon of one neuron and the dendrite of the next, and that is called an axodendrinapse. So it's axodendritic, synapse. Now, a second type would be where the synaptic knob is attached to the soma or cell body, and that is called an axosomatic synapse, this area here. Now, remember that the impulse travels in only one direction, and the impulse travels the axon to the synaptic knob, and this is where the synapse is between these two nerve fibers. Okay, so this is the direction of the impulse. It has not reached the synapse yet, so this is called the presynaptic axon or nerve fiber. And after the synapse, this is all considered postsynaptic. Okay, so you have really two nerve fibers that are connected, that are sending impulses to this neuron, okay, one direction. And this is all postsynaptic, and the impulse only travels in one direction. So you have presynaptic is the sender, postsynaptic is the receiver. Remember that the two don't really touch. Our models in the lab, and in this picture it appears that they touch, but they don't really. There's a gap in between them that we call the synaptic cleft, and here's the synaptic cleft right here. Okay, so this is the presynaptic nerve fiber, and this is the direction of the nerve impulse, and here's the synaptic knob. As an action potential reaches the synaptic knob, you have calcium channels open, and the calcium rushes in, and when the calcium rushes in, it allows for the synaptic vesicles to release their neurotransmitter. Now the neurotransmitters are released by what process here? What's happening here? Yes, exocytosis. So the synaptic vesicle moves toward the edge of the membrane of the synaptic knob and releases its contents after the influx of calcium. If calcium levels are too low, you don't have enough calcium, then it decreases the amount of neurotransmitters that's released, and this can cause some pretty serious problems. Now here's that synaptic cleft that we were talking about. It's filled with a conductive gel, like bioelectrical conductive, and also it also allows the transmission or the transfer of this chemical through it to reach the postsynaptic membrane and do its thing. Okay, so the nerve impulse reaches the axonal terminal, as we said before, of the presynaptic neuron, causing depolarization of the synaptic knob, which causes the calcium channels to open, and then the release of the neurotransmitter by the synaptic knob. So again, this is presynaptic and this is postsynaptic. So the neurotransmitter is released. And so as we started here on this postsynaptic membrane, we have resting membrane potential. But the neurotransmitter is released into the synaptic cleft. And on the surface of this postsynaptic membrane, there are going to be receptors that will bind with these neurotransmitters, this chemical, and allows for something to happen or not happen. It may block the potential for the depolarization of this membrane, or it may cause the membrane to become hyperpolarized, which would take it to further from action potential. So it depends on the type of neurotransmitter that is released. Let's look at this video. I'm not real sure if the audio will work, but if it does not, then we'll just read the captions.
Action potentials arriving at the presynaptic terminal cause voltage-gated calcium ion channels to open. Calcium ions diffuse into the cell and cause synaptic vesicles to release acetylcholine, a neurotransmitter molecule. Acetylcholine molecules diffuse from the presynaptic terminal across the synaptic cleft and bind to their receptor sites on the ligand-gated sodium ion channels. This causes the ligand-gated sodium ion channels to open, and sodium ions diffuse into the cell, making the membrane potential more positive. If the membrane potential reaches threshold level, an action potential will be produced.
Okay, so we talked about this before when we talked about excitatory and inhibitory neurotransmitter. Well, excitatory postsynaptic potential occurs when an excitatory neurotransmitter is released. The change in the potential can be graded. Remember that only an area, you have the dendrites and the soma. When you have a synapse there, the change is going to be graded. It depolarizes the membrane of a postsynaptic neuron because it is excitatory. So excitatory takes it closer to action potential. So depolarization happens with an excitatory postsynaptic potential. Now, inhibitory postsynaptic potential, those neurotransmitters that act as inhibitors, also give a graded response where the membrane is hyperpolarized. Remember, that allows for the inside of the membrane to be more negative, and on our number line, remember the negative 90 millivolts, it can become very hyperpolarized, and that will take it further away from the possibility of an action potential, so action potential of the postsynaptic neuron becomes less likely. There are both excitatories and inhibitories released at the same time. They're added together, and the process is called summation. So when summation happens, it leads to a greater potential for action potential if you have more excitatories released. If you have more inhibitories released, then it's going to be less likely of an action potential. So it's the amount of either or that determines what's going to happen, whether you're going to sum to at the axonal helix so that you have an action potential generated, or that area of that membrane is hyperpolarized, and with the summation what remains hyperpolarized and takes it further away from an action potential. So both happens at the same time. It's determined by the sum of these two together.
There are at least 30 different neurotransmitters produced by the central nervous system. We've seen acetylcholine before. It's involved in the control of skeletal muscle actions. We know that it stimulates, that's in the central nervous system, and in the peripheral nervous system it stimulates the muscle contraction at the neuromuscular junction. It may excite or inhibit in the autonomic nervous system synapses. There are a type of neurotransmitters that are monoamines. These are modified amino acids. They're distributed throughout the brain. They play a role in emotional behavior, circadian rhythm. These are present in some motor neurons of the autonomic nervous system. Some of the examples are epinephrine and norepinephrine, and norepinephrine creates a sense of feeling good. Low levels may lead to depression. In the peripheral nervous system, it may excite or inhibit some autonomic nervous system actions depending on the receptors. Dopamine creates a sense of feeling good. Deficiency may be associated with Parkinson's disease. In the peripheral nervous system, it limits some actions of the peripheral nervous system, autonomic nervous system. It can be excitatory or inhibitory depending on the receptors. Now, as you read these, several say it depends on the receptor. So wherever that neuron is located may have different receptors, and depending on the receptors there, it would make it more excitatory or inhibitory. Serotonin, it's primarily inhibitory. It leads to sleepiness. It's also enhanced by selective serotonin reuptake inhibitor drugs. Histamine, released by the hypothalamus, promotes alertness, and that's in the central nervous system. And then there's some unmodified amino acid, glutamate, aspartate. GABA, gamma aminobutyric acid, and glycine. Some are inhibitory and some are excitatory. Then you have neuropeptides. Neuropeptides are synthesized by the central nervous system neurons, and they act as neurotransmitters, but sometimes they're neuromodulators by either altering the neuron's response to a neurotransmitter or even blocking the release of the neurotransmitters. They will include the endorphins and ecophalines. Now, if you see here the substance P, it causes pain perception. The endorphins and encaphalines are generally inhibitory. They reduce pain by inhibiting the release of substance P. These encaphalines are increased during painful stress, and so in painful stress, it blocks substance P release that makes you more aware of pain. So it's almost like a natural pain reliever. It binds to some receptors in the brain and gives sort of a narcotic or morphine effect to relieve pain. So it's the body's natural pain reliever. Endorphins have a longer, more potent, lasting effect. And you have what is called beta endorphins, and remember that term. Beta endorphins relieve pain sensations that are potent, and it is potent and it has a long time effect. So relieves pain sensation, potent and long effect for the beta endorphins that are included in that group. Now, what happens to these neurotransmitters as they are released into the synaptic cleft and are bound to those receptors that determine whether it's going to be sometimes excitatory or inhibitory? You don't need overstimulation of that cell membrane, so an enzyme is released that helps to break down the neurotransmitter. And we talked about acetylcholine esterase before. It breaks down the acetylcholine so that you can have reuptake. The neurotransmitter is transported back into the presynaptic knob, and then it comes together again in the synaptic vesicle to form more. The example was acetylcholine. Was when it's taken back into the synaptic vesicle, it is acetic acid, and it binds with choline to make acetylcholine. Both above processes prevent continual stimulation of the postsynaptic membrane. That's very important. So the destruction of the neurotransmitter and the reuptake of the neurotransmitter is very important. Impulse processing. You have neuronal pools. These are groups of interneurons that make synaptic connections with each other. Interneurons work together to perform a common function. These pools receive inputs from other neurons, and each of these pools also generate output to other neurons. So it's just a process of transferring a nerve impulse from one group to the next. One type would be convergent, where the neurons receives input from several neurons at the same time. We saw the picture where you have the neurons, the synaptic knob attached to the presynaptic came into the dendrite or the cell body or soma, and so you have several neurons attached, and this would be convergence because this information is coming from two different neurons and it is received by one. The incoming impulses represent information from several types of sensory receptors. It allows the nervous system to collect, then process, and then respond to the information. It makes it possible for a neuron to sum impulses from different sources. Divergence is just the opposite. One neuron sends impulses to several neurons at the same time. It can amplify the impulse. Now, let me say this real quick. I think I've said it previously, and when we were talking about the depolarization of the membrane and repolarization, we said that the soma and the dendrites have graded responses. If it reaches action potential, the graded responses loses intensity as it travels a distance. An action potential generated on an axon does not lose its strength with distance traveled. So the impulse intensity remains the same on these axons. And that impulse can or may be amplified to activate enough motor units needed for muscle contraction, for an example.